coming. It's going to be we'll see if we have to use that. So Debbie asked me to talk about, NFSTC asked me to talk about the interpretation of YSTR mixtures and statistical applications. And I initially said, no, I'm not going to do that. So, so what I did was I promised that, because quite honestly, YSTR mixtures I would characterize as a work in progress. I think that's how I would characterize YSTR mixtures. And I see a lot of disappointed faces around here. Could I just first of all ask the audience, how many people in here are actually using YSTRs in real life, so to speak? Okay, that's a reasonable number. And how many of those, so it's about 2/3 maybe, the audience, how many people actually have come across YSTR mixtures? It's the same hand, right? Now, is that a surprise to forensic scientists? We come across mixtures. Okay, so what I'm going to do then is I didn't put this slide in here. Someone put this slide in here. Okay. Okay. That threw me off a little bit there, that slide. So what I'm going to talk about is since I've got a reasonable period of time, is to talk about Y chromosome biology for those, because the reason I'm talking about Y chromosome biology, there are some important biological consequences that affect mixture interpretation. Then I want to talk about YSTRs in casework, including statistics, use of the National YSTR database, and then talk about potential solutions to the YSTR mixture analysis problem. And as I said, the caveat is that it's a work in progress, and certainly I would characterize YSTR mixture analysis at this time as there's no general consensus throughout the community as to how to actually perform the mixture analysis. Now, when I say that, because one will be misquoted now, right? Remember, there are three things that one does in a mixture. One is to determine the presence of the mixture. The second thing is then to infer the genotype, right? Genotypes. And the third thing is to apply the stats. I think the first two are relatively straightforward, as we'll see. You know, is there a mixture present? And secondly, can we infer the genotypes? In this case, the haplotypes. These are relatively straightforward, and there are tools available for it. It's the third part, the statistical approach, which is which is where where basically it's a work in progress. Okay, enough said about that. So let's talk about some Y chromosome biology. So let's talk about, first of all, the Y chromosome. What is the function of the Y chromosome? Well, historically, we all know what the function of the Y chromosome is now, but anyway, historically, because of aneuploidies of the X and Y and sex-reversed humans, it became clear that the Y chromosome is very important for sex determination. So, for example, in Klinefelter syndrome, individuals who have 47 chromosomes, 2 X's and a Y, they were males. Or even other Klinefelters, that's the common Klinefelters, other Klinefelters with 48 chromosomes, triple X and 1 Y, 4 X's and 1 Y, 5 X's and 1 Y, they were always all males. Then when you looked at Turner syndrome, where you had 45 chromosomes with a missing Y, an XO, these individuals were females. Or if you had a triple X karyotype, 3 X's with no Y, 47 chromosomes, that person was a normal, whatever normal means, female. And 47 X with 2 Y's karyotype had a normal male. So that's one side of the equation. And the other, so that indicates that the Y is important for maleness, so to speak. And then you had sex-reversed individuals. Some individuals who had XY, so-called XY karyotypically. In other words, there was an X and Y chromosome present. Some XY individuals were found to be female, but in closer inspection, there was a small part of the Y chromosome missing. Okay, and similarly, there are XX males who have 2 X's. So you'd think they were female, but they were actually male because what they had on that X, if you look carefully, one of the X's was a small portion of the Y chromosome. And that portion of the Y chromosome was called TDF, testes determining factor. Okay? So it turned out that it wasn't just the Y chromosome, it was a small part of the Y chromosome that was important for maleness. Anyway, sex determination now in mammals is quite well understood, and there's a master gene, the SRY gene, which is present. That TDF ended up being the SRY gene. And the SRY gene in development, if the zygote has an SRY gene present, then testes development takes place. That results in the presence of testosterone and then external male genitalia. What the other consequences of testes development is that you have Mullerian inhibiting factor and Mullerian duct atrophies. In other words, the physiological structures that would normally result in female phenotype are atrophied, and so you end up with maleness. So the bottom line is in mammals, and meaning humans in particular now, the default gender is female, and if you have the TDF gene, that will cause in the zygote the production of a male individual. Okay, so the Y chromosome, here's a classic view of the Y chromosome. And you have the TDF master gene, which is present on the, can't do it. Let me try this. The TDF master gene is present on the Y chromosome. And I just referred to what that does. And of course, the Y chromosome, there's going to be patrilineal inheritance that will be passed unchanged from father to son. We'll talk about that. There's no recombination in a large part, most of the Y chromosome. But there has to be some recombination because during meiosis, the Y, during the formation of zygotes, sperm or egg, you have to have, the chromosomes have to be properly distributed into the various zygotes, into the various germ cells. So on the ends of the chromosome, you have the pseudo-autosomal region, PAR, on the short arm and the long arm. And these are the regions that recombine with the X. So there are homologies at both ends of the chromosome that recombine with the X. But the vast majority of the chromosome is called the NRY, non-recombination region of the Y chromosome. And that's the vast majority. But when you again look at the classic view of the Y chromosome, you have euchromatin here, which is where all the functional genes are. Not that there are a large number of them, but there are some. And then you have heterochromatin, which is a region of the chromosome that basically has no known function at this moment, no known physiological function. But of course, it may have structural, there may be no known genic function, but there may be structural reasons why it's still present. And so the concept always was that the Y chromosome is junk-rich and gene-poor. That was the classic view. But from our standpoint in forensics, what we have to realize is that the Y chromosome evolved from a common set of autosomal chromosomes that both the X and the Y have a common origin. and this is a representation of what is thought to have occurred, is that you have a pair of autosomal chromosomes early on in evolution. Somehow one of these two chromosomes mutated or had the formation of a sex-determining allele, SDA called here, and there were sufficient differences in that, some rearrangement, some mutations that prevented recombination taking place in this region. You have suppression of recombination, and you then have, and over a long period of time, this extended further along the chromosome. But as we'll see, there's been a lot of transposition of actual sequences from the X to the Y, etc. And that's why we have some problems occasionally with determining or designing y-STRs that are unique to the Y chromosome and are not present on the X. So anyway, so you have this recombination suppressed chromosome Y. You then have chromosome divergence through time, and then the chromosomes begin to differentiate, and because the Y chromosome is only believed to be required for sex determination and a few fertilization type of genes, the chromosome shrunk in time, the Y chromosome. And so what we have is a heteromorphic pair now, instead of this original situation where both chromosomes were the same. We now have the X and Y chromosome. And some people believe that although this is now, I think some people, more people than didn't believe that the Y chromosome could in fact disappear. I think that over time, but I think in the light of a lot of recent work, people are doubting that hypothesis, and it's a hard one to prove. It's the future, right? So it's but there is a theoretical possibility that Y chromosome will just disappear, right? So you may ask, what does that mean? So, and I ask the same question. So, but when you look in more detail, so that's how the X and the Y chromosome then basically evolved from a common ancestor. So you would imagine then that there's common sequences. You'd think there was some remnants of that evolutionary past. Of course there is. That's how we discovered that there's an evolutionary past. So there is some evolutionary relatedness. But after the human genome was sequenced, people turned their attention to the Y chromosome, and that was sequenced also. The original first draft of the human genome had a certain amount of Y chromosome sequence. More work had to be put into it. A couple of years later, a draft of the Y chromosome sequence was published. And in fact, even to this day, it's not all sequenced, because that heterochromatin region is intractable to a lot of standard sequencing methodology. But anyway, when you look at the Y chromosome in some detail, shown in A here, you have here's a centromere here. So you have the short arm, YP, and YQ is here. And what this diagram is showing is the different types of class of sequence that make up the euchromatin region. And as you can see, there are several classes here. There's an X transposed class in pink. There's an X degenerate class, and there's an ampliconic class. And I'm ignoring the heterochromatin, which is in this Q arm, because we don't know what it does and we don't use it for anything in our business. And there's a pseudo-autosomal region, right? So the three classes, there's X transposed class. The X transposed class is a whole region of the X chromosome has just been transferred in two blocks, straight into the Y chromosome. So there are regions of the Y chromosome that are basically the same as the X. It's about 99% homologous, right? That's one whole class, and that's in the pink here. You see, in this short arm here, pink, and here, and then we have another series of structure classes, sequence classes, in yellow here, the X degenerate classes. And these are another set of sequences that have similarities to the X, but they're now sufficiently different that they've, over a period of evolutionary time, they've basically changed. Okay. It turns out, for example, that the sex determining gene SRY, which is present in which is the testes determining factor, that gene is actually present in females, right? But it's part of the X degenerate class. So there's another gene in females called SOX3 that has a similarity in females. When I see females, also males, on the X chromosome. But that gene has a different function, different structure, and it's diverged in terms of what its function is. Okay. Anyway, so these X degenerate sequences, there's some similarities, but a lot of differences. And then we come to the so-called ampliconic sequences. And they've used this term ampliconic. It's got nothing to do with amplicons that amplicons to us are amplicons, PCR products. The ampliconic sequences here refer to a strange set of sequence, has a strange set of sequence characteristics. They're palindromic sequences, large numbers of them, very large in length, and they're palindromic sequences. And the degree of sequence homology is extremely high, 99.9% or more. So when you look at these ampliconic sequences, you'll find say a sequence of several million bases in one, or several 100,000 bases in one region of the chromosome. Further along the chromosome, you'll find another sequence with the exact same sequence, 99.9%. In fact, the sequence is so close that it's impossible to have been maintained that way through evolution. In other words, it could not have been maintained that way in the absence of some force that the organism uses to maintain that sequence identity. It's something like gene conversion or some other mechanism. And it's unknown why there's such great conservation there. Because typically, as you know, if you have genes or structures that even have some of them are genic, but some have no known function, they can evolve over a period of time just due to random drift, right? Well, in this case, they don't. So they're very, very high sequence complementarity. Anyway, the whole point of going through this and into details to let you know that the X and the Y are very, very, there's a lot of homology. And so when YSTRs were being designed, many loci which could have been used have to be rejected, because when you first tested the YSTR, some of them are also present on the X, right? So that's a problem. It may not be the STR itself, but we don't detect it. We're not looking at the STR. We're looking at the unique sequences flanking it. So sometimes you're looking at a unique sequence. You find a set of primers. They don't work, or they're present in the X. So we have to redesign new primers. And it's very difficult sometimes to arrange it such that the sequence primers are unique to the Y chromosome. So in all the validation studies, when we do Y chromosome analysis early on in YSTR development, a lot of people spend a lot of time taking their samples and testing them with female DNA, with large quantities of female DNA, because obviously it's under those circumstances when there's a lot of female DNA in a male, and there's a male present, that we want to use YSTRs. And so you'll see that a lot of people have done that, and even to this day, you may occasionally still find cross-reactivity, depending if something's going wrong with your PCR. If you reduce the stringency of your PCR reaction for whatever reason, and it can be done because of a malfunction of your equipment, but there are other things, other additives to that PCR that can alter the melting temperatures, etc., of your primers that can affect and therefore cause mispriming, etc. Anyway, that's probably more than you wanted to know about the Y chromosome, but I'm trying to make a point that absolutely the relatedness is very important. We have to understand that. Okay. Now, during meiosis, here's basic biology. On the left is meiosis, on the right is normal cell division. Obviously, during meiosis, meiosis, of course, produces the germ cells, so sperm in males and the egg in females. And what happens is that the whole point of that is to produce the gametes. So there's a reductive division. And so what happens is in the cells that make up the meiotic cells, they have to replicate their DNA. The chromosomes, so in other words, the chromosome 1 will have to be replicated to form 2 chromosome 1s, etc. The chromosomes pair and recombination takes place, right? Gene shuffling, so to speak. And then you have cell division 1, cell division 2, etc., and you form the gametes. So that recombination event is where the genes are shuffled. So when we're looking at the mutations that take place, when we look at variation that takes place, say in the Y on any chromosome, let's forget the Y just now, on any chromosome, basically there will be gene shuffling taking place. So you'll have fragments of one, from the paternal chromosome will be juxtaposed to the other chromosome. The maternal and paternal chromosomes will have recombined. Now the difference is with the Y chromosome is that despite the fact there's evolutionary similarities in some of the X sequences, the X transpose sequences, there's no known recombination that takes place. There must be a suppression of that recombination in that NR-Y region. But at the pseudo-autosomal regions, in both ends, but particularly at one end, the P arm, you have that recombination taking place. So there will be gene shuffling, if you want to call it that, in one part of the Y chromosome, but we're not interested in that from our perspective. Okay, so basically, what happens is that the only force for change then in a Y chromosome environment is not gene shuffling, like as with the autosomals, right? The only way that we can get change in the population through mutation, right? And obviously, depending on which sequence we're looking at, the mutation rate will change. So STRs, right, which is what we're interested in, they have a high mutation rate as opposed to a single nucleotide polymorphism, which is a much lower mutation rate. So one could be in the order of 1,000 to 10,000. That would be the STR, whereas a single nucleotide polymorphism would occur, the frequency of a mutation at SNP would be 10 to the 8, that sort of number. So there's a big difference. Okay, so in other words, the Y chromosome then is passed en bloc then. Like it's now a linked series of markers basically from father to son. So here's the typical Y-linked inheritance. At the top you have the father and the mother. In the black is the male ancestor, and of course you can see that there's mating takes place, and there are several children. There's two male sons, obviously male sons, two sons, and two daughters. And obviously the males will pick up the Y chromosome type, females don't. And then of course these two males mate, and the first one to the left has two sons, and they both have the same Y haplotype, Y chromosome type, as the ancestor. Whereas on the right-hand side, of course, the mating has obviously a son and a daughter, and only the son gets the Y chromosome, obviously. Okay, so that's as much biology as I think we can stomach here, right, to set the scene as to what we're doing with Y chromosome analysis. So the reasons why we are interested in Y chromosome analysis from the forensic standpoint is that, quite honestly, there's a genetic determinant of criminality. There's an environmental component, but the Y chromosome is correlated with criminality, right? So, because 80% of all violent crime is committed by males and 95% of all sexual offences, and I think it's statistically significant when you take into account the environmental aspects. So that's why we're interested. A lot of males commit crimes, and they have the Y chromosome. But of course, it's only under certain circumstances that we would employ as forensic scientists, why we'd employ y-STRs as opposed to standard DNA testing. Typically, it occurs when you're trying to determine the genetic profile of a male donor in a female DNA admixture, where the female to male ratio is very, very high. I've said more than 20 to 1, but it's often much more than that, 1,000 to 1. And autosomal analysis fails, right? Or is not informative or not possible, right? So obviously sexual assault cases, where there's saliva-saliva interactions, you can't do a differential then. Saliva-vaginal secretions, same thing. Extended interval post-coital samples is another instance, because the sperm are very, they may be premature lysis of the sperm into the non-sperm fraction because of their fragility, etc. And you could have aspermic males, oligospermic males, although of course nowadays it's quite easy to obtain a standard autosomal profile from such individuals, but Y can also help on those occasions where you don't get a good result from aspermic individuals. And obviously normal degradation and loss over time. Well, that's another reason. You could also use Y chromosome analysis for no need for differential extraction, and that was thought to be early on. It may be of benefit. We don't have to do a differential extraction now. That would be in disagreement with the SWGDAM the quality assurance standards. So it's probably not a good idea to do that. And what we found in others, in fact, is we've found no benefit in using y-STR analysis without differential. We've always found in our hands now, despite earlier work, and others have confirmed this, I believe, that the differential extraction is still the best way to go anyway. Obviously, Y chromosome analysis could be used to help in aiding the number of semen donors. So if you have a mixture, then if you do a Y, then you'll be able to help determine the number of semen donors or male donors, right? So you see a mixture of 3 individuals. You think 3 individuals on a not an intimate sample, because it's but on a stain, and you think, you know, it could be 1 female and 2 males. Well, if it's 3 males, then you may see 3 males clearly, but it's going to be easier to look at the number of bands, because obviously you'd expect in the Y chromosome from a single individual you get one allele, depending on the locus, but 1 allele. So if there's a mixture of 2 people, you expect 2 alleles. Right? A mixture of 3 people, you get 3 alleles, as opposed to the autosomal situation, as you know, where it's more complicated. Missing persons cases, of course, it can be used in criminal paternity, disaster victim ID, etc. It can also be used to provide additional statistical discrimination. You may have a partial profile with the autosomal, and you want more certainty about whether or not this person is still included, and so you can do y-STRs and get more statistical information, and then apply it to the autosomal situation. We've not talked about y-SNPs. Y-SNPs may be useful in the future for various things, including ethnogeographic ancestry prediction from the paternal line. But also, lastly, and it's coming, and we just heard about it this morning, that Virginia's got a familial searching program. Familial searching, y-STRs can play a major role in familial searching, right? Because obviously when you do familial searching, you get a list of candidates, especially if you're using large databases, you may have a large number of candidates. And of course, the vast majority of these candidates are false, meaning they are not related to the crime scene sample. And obviously, if you use y-STRs, you're going to eliminate 99.99% of falsely accused or falsely associated males if they're not associated with that crime. So I think it's going to take. So I think the Y chromosome analysis, y-STR analysis, will play a role in that area as well. I should also say, actually, just because it just occurred to me to say this, that there's been some recent advances in y-STRs. So we have a standard set of y-STRs at the moment, say Y-filer, etc., 17 loci. But a new set of loci have been recently described by Manfred Kaiser and his group, where they're highly mutating y-STRs. Because when you look at all the y-STRs, there are hundreds of them, right? And when you look at the mutation rates vary. And what Manfred Kaiser has done is identified a number of highly mutating y-STRs. Very high mutation rate, higher than the average. So why is that of any interest? Well, we often want to be able to differentiate. I have a case myself right now, right, that I'm involved in, where there are 3 individuals who are persons of interest in a case. And the first individual is the grandfather, the second individual is the father, and the other one's the son. They're all persons of interest. And so they all have the same y-STR haplotype, unfortunately, for this case. The question would be, but can we still differentiate these? Because it's only Y, there's no autosomal in this case. So the question is, could we identify these individuals? If you have highly mutating y-STRs, there's a probability, a discrete probability, and it can be measured, that you can in fact show a single mutational difference between the father and the son. Right? Now, there's no question that I have 2 children, 2 male children. There's absolutely no question in my mind that our Y chromosomes are unique. If you were to sequence my son's Y chromosome and my Y chromosome, there will be differences. But they're private mutations. But they will be there. We don't have the technology yet to find them, but we will find them one day. But in the meantime, we can maybe look at these highly mutating y-STRs and use them in those cases where you are trying to distinguish the individuals in that male lineage. Anyway, so my whole point in saying this is it's not totally dead, the subject. There's still quite a lot of work going on. Okay, now it's also not new either, y-STRs, although it would seem that way occasionally when you talk to some groups. But 1997, Kaiser et al. Produced a paper that was based upon a study that a European group got together and like all good forensic scientists, sat down in a room and decided that we're going to test a few y-STRs. And they came up with after a study, they came up with what was called the minimal haplotype. And it's a European group, the minimal haplotype, 1997. So these were a set of loci that were recommended for potential casework use in 1997. Then but there was a very limited number of loci. But then there was a couple of papers in 1999 and 2000, White et al. And Ayub et al. Producing novel YSTRs that opened up the sort of our vista, and we said, okay, we could now get a reasonable number of YSTR loci. Okay, and what happened was the first commercial kit was released in 2001, and that was YPlex 6 by Ballentyne. That was the first kit, and that was based upon the minimal haplotype loci. The same time, that same year, New York City OCME was the first lab in the US to start reporting YSTRs. So New York City OCME have been doing YSTRs since 2001 in actual cases. Then in 2002, Kaiser et al. Published another a paper that further expanded the number of loci to well over 100 loci that we could choose from YSTRs. Then the YPlex 5 kit came out from Ballentyne, which is a set of additional markers that now came from some of these newer markers. And that was YPlex 5 in 2002. And then in 2003, Ballentyne put them all together into one kit, and that's the YPlex 12. And so some of these older cases are done using the YPlex 12 or YPlex 6 or 5. So we will come across them occasionally. In fact, my own lab, when Ballentyne decided to basically close down, they donated to our lab some kits, some of the Ballentyne YPlex kits, and to this day we still use them for training and for classes, etc. And they're perfectly fine. They work great, and they must be six years old or so. Okay, so don't tell Applied Biosystems that the expiration date can be years away from. Okay, but the YPlex, that's now that technology is no longer available because of various commercial transactions that went on, because Ballentyne no longer exists. And so, but anyway, in 2003, PowerPlex Y was released from Promega. In 2004, Y-Filer from Applied Biosystems, and we haven't really progressed a lot beyond that. There are some kits available from other companies in Europe, but they use the same loci for the most part. Okay, so the minimal haplotype loci are listed here. That was the listing by Kaiser in 1997, first used in Europe, and formed the basis for all multiplex development in the US. Then SWGDAM got together, and I was the chair of that committee at SWGDAM, and we recommended in 2003 now the minimal haplotype loci plus a couple of extras, 438 and 439. And I've already told you about the commercial kits at the moment. So I'm going to guess in this room, no, I'm not going to guess. How many people who were doing YSTRs are using Y-Filer? How many people are using the PowerPlex Y? Yeah. So again, there seems to be more people using the Y-Filer than the PowerPlex Y. I don't know what the ratio was. It could have been 5-to-1, something like that. And I'm assuming that the reason for that is because that the Y-Filer, in a large percentage of cases anyway, because Y-Filer has 17 loci as opposed to 12 in the PowerPlex. Would that be one of the main reasons for those people who are using Y-Filer versus? Now, this is not a commercial endorsement of a particular company. It's the number of loci I'm interested in, right? And I certainly know that the Promega Corporation are looking into increasing the number of loci, so as they should do. So would that be the main reason, do you think, that it's mainly for the added discrimination? Okay, because it's absolutely clear that you will get more discrimination with 17 loci than with 12. Okay. So if you look at here's a list that minimal haplotype loci are here. SWGDAM core loci, we added 3 loci. Ballentyne 12-plex comprised all of the SWGDAM loci. And Promega PowerPlex Y also contains these loci. ABI Y-Filer has these extra 5 loci. Now, just to talk about the number of loci, there's some discrepancy in the literature when you read the number of loci. I would say there are 17 loci 17 YSTR loci in the Y filer. But some people will say there's 16, and that's simply because 385, for example, you have 385, and you have two alleles there, but they're from two different separate loci, actually, and they are two separate loci, but we can't differentiate when we type this, is if it's a 12, 14, say, we don't know if 12 is from the first locus or from the second locus, but they are two separate loci. So I just as a geneticist would say they're separate loci, but so you have to be aware that people will say it's 16 loci, and some people say there's 17. I say there's 17, but so what? You know, I mean, that's it. Okay, now, when we come to, this is the wrong way, so if you look at the right-hand side, I shouldn't have done that. On the right-hand side, there was a famous football manager, soccer manager, Bill Shankly. So some people believe football, soccer, is a matter of life and death. And Bill Shankly said, I'm very disappointed with that attitude, he says, because I can assure you it's much, much more important than that. Now, that was Bill Shankly. Now, you say, why would I bother mentioning such a thing? Because as a boy growing up, I used to think that's an amazing concept, you know. But then look on the left-hand side by a very famous statistician, Balding, who many of you will know, the name anyway. He says, a detailed understanding of the influence of all the factors on the evolution of our profile proportions requires a lifetime of study and more, right? So, to me, they're both the same. These two statements are equivalent. So, the reason for saying this is that if somebody like Balding requires a lifetime of study and more, right, to understand these things, then most of the rest of us are going to be a little bit behind. So, statistics again. Now, we have some people in this room who have spent a large part of their life studying these things. So I'd be interested in what they think about David Balding's statement. So in terms of Ys then, so I've just talked about Y chromosome biology, why we use YSTRs and some, you know, what the kits are and what's available to us. So we then have to, again, so we can just use our standard methods for capillary electrophoresis separations, and we can get a profile, right? It's now a haplotype, because now they're linked loci. Now, so when we look at YSTR interpretation guidelines, then similar issues pertain to YSTRs as they do to autosomal. There's going to be a lot of similarities and then some differences, right, in the stats, etc. But in terms of the very similar general issues, there has to be thresholds for detection and interpretation. You need to have that, right? We need to have statistics for the so-called probability of a match, if you want to use that term. Mixtures, we have to talk about that, the mixtures. There's stutter, and validation studies have to be conducted in concert with guidelines. So a lot of this is quite familiar territory as a general concepts. Now, when we come to the stats, however, and I'm not talking about mixtures yet, right? Then, because you have a profile now, and we need to then attach a weight to the profile of the haplotype. Still a profile. Then estimating the rarity of that match is done differently to how we do it with autosomal DNA markers, for the obvious reason that the autosomal markers are on different chromosomes, they're independent, they've been shown to be independent of one another, and you can multiply the frequencies, right, of each from each loci together to get the overall random match probability. Obviously, because of linkage here, both physical and genetic linkage, each haplotype is treated as an allele. So in other words, your Y chromosome now, this whole region with the particular at each loci, whatever your particular allelic designation is, that loci, that overall haplotype is then what is now treated as an allele, and you know it's basically treated as an allele, and the total number of possible haplotypes comprise the alleles of a single locus. So in other words, if you take the 17 markers, then that extended haplotype is now regarded as one allele, and then in the population you see, well, okay, in the population then these are alleles. So you go into the population and you see how many times that particular combination of markers that comprise the extended haplotype is found or not found. Well, it has to be found. It's been found once at least, if you've seen it. Okay, So that composite multi-locus profile is treated as a single locus haplotype. Now, I told you there's no evidence for recombination across the majority of the Y chromosome. Now, some individuals have shown that there may be the linkage is not the linkage disequilibrium is not total, meaning that there's some evidence that pairs of loci may be somewhat independent, and that's due probably to just mutation that takes place, right? So when you have a father, you know, the haplotypes pass from father to son, and then from that son to his son, et cetera, down the line through evolutionary time, mutations will take place, right? And if we're lucky enough with these highly mutating ones, we could find it even in that one generation. But with the set I'm talking about, it will take some time, but it will mutate in time. And if there's enough of that mutation taking place, and depending on the population structure and the evolutionary history of humans, some of them may end up being somewhat independent in some populations, et cetera, but for our purposes, we are going to assume that any such non-linkage disequilibrium is of no great significance to us. So we'll still assume that there's a haplotype and that we can just measure and count in the population. So, so you cannot employ the product rule, obviously, to estimate the rarity of the Y types. So let's talk about the counting method now. The counting method is, as I said, very simple. You know, a Y haplotype, the evidence sample is compared to a reference database of individuals, hopefully unrelated. Then the number of times the Y haplotype is observed is counted. And however, the size of the database can be often limited. So we may have databases that I'll talk about, or our database that we run, it may be 18,000 profiles, but for any subpopulation in there, it may be a very small number, smallish number of people in that sample. But with databases, there are many possible haplotypes will not be observed, right? So, and there will also be sampling error. So sampling error is something we have to take into account because these are relatively small databases compared to the population. So we have to talk about sampling error, but also many possible haplotypes that could possibly exist have not been observed. And it's going to be a problem with mixtures because in mixtures, when we look to try and deconvolute a mixture, the number of haplotypes that could be present in the mixture, some of which will be observed in the database and some of which will not. Of that class that are not observed in the database, there's a problem because some of them may in fact exist in the human population. In fact, we use a formulation to describe that, as we'll talk about in a second. Right? When you find out in a case, you find a haplotype and then it's not observed in the database, we employ a formulation for that. But it's not been observed, but we can give an estimate. But in this mixture situation where you have an unobserved haplotype that's unobserved in the database, there's two classes. There's one set of haplotypes that have not been observed, and they will never be observed in the human population. You know, in terms of they've never existed and never will exist. Just because evolution will not allow it to happen. They're too far removed from present day, humans. Or it maybe take so long. But there's a whole other class, of course, that probably do exist in the population, and we can't, at this moment in time, easily differentiate. We can't just take a haplotype clearly easy. It's not easy to do, to take this unobserved haplotype and say, okay, that one exists in the human population or is likely to exist, and this other one doesn't, or could or couldn't. So in terms of the formulations, the standard counting rule is shown here at the top. P equals x over n. X is the number of observations of that haplotype, and n is the sample and the size of the database. And that database could be the total database, or it could be a subset of the database. Caucasians, for example, or it could be the total, right? In terms of the sampling error, sampling corrections, then there have been two sampling correction approaches made. The first one is shown, and that the p plus or minus 1.96, square root p1 minus p over n, that is based on an assumption of the normal distribution. The sampling distribution is normally distributed, right? And that was used in the community for some time. However, as pointed out by our good friend here, John Buckleton and Bruce Weir, and as we all know, actually, from basic statistics, the true sampling distribution here is binomial, and the normal distribution is quite a good approximation to the normal under many circumstances. But in our situation, where we're dealing with a small number of observations in a larger database, then it's not appropriate. It's not the most accurate way of doing it. In fact, it's wrong, basically, when you have a small number of observations. When I say it's wrong, it's just a model, right? Remember we said, so a more accurate way of doing this is to use the binomial distribution, right? And so to use an exact binomial 95% upper confidence interval, right? And so for this, however, we use the last formulation there, which is a formulation, it's called the Clopin-Pearson formulation. And it does require, this particular formulation requires a computer to help doing this because it's an exact test, okay? And the US y-STR database that we'll talk about uses this Clopin-Pearson method. In fact, the algorithm, the spreadsheet that's used to do the calculation came from John Buckleton. So there are alternative ways of calculating this. They may be faster, etc. But at this moment, on the US y-STR database, this is the formulation. This is the formula used. It's the 95% upper confidence interval based on the binomial distribution, and the actual computational part of it is based on an Excel spreadsheet that John provided to us. And thank you very much, John, for that.